Hello friends, this video on class 12 biology part 2 revision is brought to you by ExamFear.com. No more fear from exam. So as we had discussed in our part 1 crash course video of biology that these are the priorities of the various chapters that you have in your class 12th biology syllabus. So we had already covered the first 7 chapters in part 1. So in this part we are going to cover the remaining chapters, that is starting from biotechnology principles. So as you can see, biotechnology principles and applications, organisms and population, ecosystem, environmental issues. So these are the 5 chapters which are of medium priority. Then you have enhancement in food production and microbes in human welfare, which are of low priority. And then you have biodiversity and conservation and reproduction in organisms, which are of very low priority. So based on this priority level, you can plan your revision timetable before your board exams. Okay, so let's get started with biotechnology and principles. So in this chapter, we are going to talk about some of these important topics like restriction endonuclease, formation of recombinant DNA, competent host, PCR, that is polymerase chain reaction, PBR322, YAC vector. So these are some of the topics from where you tend to get, good number of questions. So let's quickly revise the entire chapter. Biotechnology, what do we do here? Use of biological organisms and processes to manufacture products useful to humans. So we make use of living organisms to make things which are useful to human beings. So that is biotechnology. So we are combining bio, that is life, with technology to create useful products. Now, when it comes to biotechnology, hybridization or traditional hybridization, where we kind of take desired genes from different organisms and produce a hybrid with all the desirable traits, right? But that was a traditional method which was followed. Now, what was the advantage? That we used to get organisms with our own desirable traits. But there were certain disadvantages associated with that. For example, undesirable genes also get expressed, undesirable genes also multiply. So along with the desired traits, there are certain undesired traits which also come as a part of the process. So there comes the concept of biotechnology, under which we talk about genetic engineering. So we play around with the genes, with the desired genes. So at the gene level, we are working in genetic engineering. So only desired genes are introduced into the target organism. So what exactly is genetic engineering? It is manipulation of genetic material, that is DNA, to bring about desired modifications in an organism. Now that sounds very interesting, because here we are bringing a change in the DNA itself. So whatever new organism is created, so that organism has a desired DNA, and because of which when the DNA replicates, multiplies, so the same trait gets expressed. So the question is, how does the DNA get manipulated? Now here, this entire process takes place in a few steps. For example, first of all, DNA with the desired genes is identified. Then the DNA is cut. And how do we cut the DNA? By using scissors. Now, what are these scissors? They are not the scissors that we normally use to cut papers. These are DNA scissors, which are restriction enzymes. So these are those enzymes which can cut DNA at specific locations. Now, desired cut piece of DNA is obtained. So with the help of these restriction enzymes, you cut the desired piece of DNA. Now what do? Then comes the role of vector, that is vehicle DNA molecule to transfer the cut piece of DNA into the host organism. Let's say this is the host organism's DNA, and this is the cut piece of the DNA which has the desired genes. Okay. Now what will the vector do? It will transfer this cut piece DNA into this target organism. So who acts as the vector? The plasmid DNA act as vectors. So what are plasmid DNA? So plasmid DNA are different from chromosomal DNA, and the speciality is that they are capable of self-replicating. So these plasmid DNA, they act as vectors. So as a result, what do we get? We get recombinant DNA. So as you can see here, this is the plasmid DNA in which this particular piece will come and get joined. Now, how will this get joined? With the help of enzyme DNA ligase. So it helps in the linkage. So it acts as a glue so that it can stick that cut piece DNA with the plasmid DNA. Now, once the cut piece DNA is linked with the plasmid DNA, what we get is called the rDNA or recombinant DNA. Okay, so now once the recombinant DNA is formed, then this recombinant DNA is transferred to the host organism. Now, once this is transferred to the host cell, now this recombinant DNA can create multiple copies using enzyme DNA polymerase of the host cell. Now, anyways, this vector is basically a plasmid DNA, but in that plasmid DNA, you are introducing your desired set of gene. Now, the DNA in itself was capable of producing multiple copies, which it will anyways do. But now when it produces multiple copies inside the body of the host cell, all those copies will have the desired traits. So an organism and its progeny can be genetically modified. So in this fashion, we are not only modifying that particular organism, not only the host organism, but the entire future generations of the host organism, because we have modified the DNA, right? And from that DNA only the future generations will get their traits. So in this fashion, using genetic engineering, we can bring modifications not only to one particular organism but to an entire generation. Now, genetic modification of an organism. What is it? Identification of DNA with desired genes, extraction of piece of DNA with desired genes, as I said, with the help of the restriction enzymes, formation of recombinant DNA, introduction of recombinant DNA into host, multiplication of recombinant DNA in host, transfer of recombinant DNA to progeny. So if you look at this picture, what do you see? The peel looks like that of a banana, but inside you have a corn. So it is like a combination of banana and corn. Similarly, if you look at this is your cotton, the normal cotton flower, and the other one is a BT cotton. What is that BT cotton? BT cotton is again genetically modified cotton, where the gene is modified in such a way that it is more resistant to pests. So that means it will have a longer life, its productivity will be more, so it is like a more desired thing for human beings. So that is BT cotton. So in this fashion, we genetically modify different organisms. Now, what are the important tools for genetic engineering? Genetic engineering is also known as recombinant DNA technology, because here the main part of genetic engineering is that recombinant DNA is formed. So the important tools are restriction enzymes, which act as scissors to cut DNA, vectors, they transfer DNA, ligase enzymes, they act as glue to fix the cut DNA to the plasmid host organism, because inside the host organism only we put the recombinant DNA, and polymerase enzymes, they help these recombinant DNA to multiply inside the host cell. The restriction enzymes. So DNA cutting enzymes found in bacteria, they cut DNA at specific sites. So that is their speciality. They belong to a class of enzymes called nucleases. Now there are two types of nucleases, exonuclease and endonuclease. Exo means outside. So exonuclease removes nucleotides from ends of DNA, from the outside part. And endonuclease removes nucleotides from specific positions within DNA. So when we have to remove nucleotides from inside the DNA, that time endonuclease. When we want to remove nucleotide from one end, like towards one end, we want to remove a few nucleotides, then it is exonuclease. Now, recognition sequence of restriction endonucleases. So each now how does this restriction endonuclease work? Because we are saying that they can cut DNA at specific locations. So how will they cut DNA at specific location? Because at a specific location, they recognize a specific sequence of base pairs. So whenever they come across that specific sequence of base pairs, their cutting action starts. And that specific sequence is called recognition sequence. For example, let's talk about some restriction endonuclease enzymes. One enzyme is BAMH1. So this enzyme, this enzyme always looks for a specific sequence. If it finds that sequence, then it breaks the bond between GG, that is between two guanine. And what is that? So let's say that if this is the pattern, so what is the sequence that it looks for? This is the sequence GGATCC. So whenever it comes across this sequence, it breaks the bond between GG. Similarly, it will break the bond between GG because we have two complementary strands in a DNA, right? So as a result, it breaks it in this fashion. So this entire sequence gets broken into two parts like this. Similarly, if you talk about another restriction endonuclease like EcoRI, so this for this the sequence is GAATTC. So for every restriction endonuclease, there is a different recognition sequence. So for BAM H1, this is the sequence. For EcoRI, this is the sequence. So whenever a specific sequence is encountered, then that restriction nuclease which can react to that specific recognition sequence will respond. So in this case, again, it breaks the bond between GA and GA. So this side also it breaks the bond between G and A. This side also it breaks the bond between G and A. And as a result, you get these two parts of the sequence. So each restriction endonuclease looks for its specific recognition sequence in the DNA. So let us look at the stepwise working of the EcoRI enzyme. So EcoR1 recognizes its specific recognition sequence, as I said, and the sequence is GAATTC. Then it binds to the DNA, as you can see here. Cut the two strands of DNA at specific points, like it cuts the bond between GA and GA, this side. Cut between the same bases on each strand. So this is one strand between GA. Similarly, on the other strand also between GA. DNA pieces are formed with single-stranded ends, that is sticky ends. So if you see here, you get two parts of DNA and these parts will have sticky ends, right? So this is how the restriction endonuclease actually works. So now let's talk about recombinant DNA formation. So as we have learned that how is a recombinant DNA formed, cut piece DNA and vector DNA, they combine to form the recombinant DNA, right? So now this EcoRI will recognize the sequence GAATTC, then it will cut between the bases G and A on both the strands. So you get two pieces. For example, here you can see that this was the entire thing and now you got the two pieces. Now each of these two pieces will have sticky ends and these sticky ends are joined by DNA ligase, like as you can see here. So the green part shows the vector DNA, the blue part shows the foreign DNA, or basically the desired DNA from where a cut piece is obtained. And in this fashion, both of them join together to form the recombinant DNA. So you often get questions on recombinant DNA technology. So you should be aware of the functions of restriction endonucleases, DNA ligase, DNA polymerase. So you should know their functions. Okay, so that was the recombinant DNA technology. As you can see here, the foreign gene, small part, this is plasmid, which acts as the vector DNA. They both combine to form the recombinant DNA. Now, one important point to be noted is that the same enzyme should cut vector DNA and foreign DNA for the formation of recombinant DNA. Why? That's because if the same enzyme cuts them, only then their sticky ends will be able to combine with each other. So it is important that the same enzyme, so let's say that the same restriction endonuclease should cut both of these DNAs, right? So now this final recombinant DNA that we obtain is our gene of interest, because this is the gene that we wanted, and now this is also capable of replication because it is kind of added to the plasmid DNA. So it is capable of replication as well. So they are the specialities of the recombinant DNA. Now let's talk about cloning vectors, some of the examples at least. So what are those things which can act as vector DNA? So one is plasmid, that is circulate DNA that replicates autonomously inside bacterial cell, that is its speciality. Bacteriophages, viruses that multiply inside bacterial cells and they high copy numbers. So they have very high copy numbers. Cosmids circulate DNA with features of both plasmids and phages. So they are like between both the two. Some characteristics of plasmids, some characteristics of bacteriophages. So it's like a combination of the two. Now when we talk about cloning into a vector, it is possible only if the vector has the following features. For example, it should have an origin of replication. We need a selectable marker at unique restriction sites. Now, the main idea is that the foreign gene must be able to replicate inside the host. Now, that is very important. So, there are two things. Let's say that this is a vector and we add the cut piece to this vector and we form the recombinant DNA. Now this should be, after entering into the host cell, should be able to replicate. That is very, very important. So now when we talk about origin of replication, what it is it means that the process of replication starts from a specific sequence on DNA, which is called the origin of replication, and it controls the copy number of linked DNA. So the first widely used vector in genetic engineering was PBR322. So how did we get this name? P stands for plasmid, BR stands for Bolivar Rodriguez, who first identified this vector that was used for genetic engineering. And then you see that there are various sections. So like the blue part is denoted by ORI, which means the origin of replication. So this is that area from where the process of replication starts. Then we have the restriction sites. For example, if this is the vector, now there are different sites for different restriction enzyme to act. So specific recognition sites need to be present on the vector for each restriction enzyme. So you see for SAL1, this is the site. For BAM-H1, this is the site. EcoRI, this is the site. So for different enzymes, there are different sites from where they would start working. Because we have learned, right, that these restriction endonuclease enzymes, they recognize the sequence and then they cut that specific piece of DNA. So when we talk about selectable marker, what does that mean? That means antibiotic resistant genes. For example, tetracycline, ampicillin, chloroamphenicol, kanamycin. So these are the various regions for which are antibiotic resistant. Now, how does this selectable marker help? They help to identify and distinguish between transformants and non-transformants. Now, I am not getting into the detail of all of these because we have already explained this in the detailed video. So, if you really want to understand this in detail, so please, so let us now quickly discuss about competent host. Now, another important thing is that the DNA can't pass through the cell membrane of the host cell directly. So it is important that the host cell is made competent to take up the DNA, because this is like, as you see here, the yellow portion shows your desired gene, that is the foreign gene. The red portion is your vector. So overall, this red and yellow thing is your recombinant DNA. Now for this recombinant DNA to enter into the host cell has to be competent to take up that DNA. Now why do we talk about that? Because when you look at the membrane of the host cell, the cell membrane has a hydrophilic and a hydrophobic end, as you see in this case. So therefore, it is important that the host is made competent so that the recombinant DNA can enter inside the host cell. So again, competent host is another important topic from your exam perspective from this chapter. So it is important that you learn about this topic in more detail from the detailed videos. Okay, so there are several techniques to insert the recombinant DNA into the host cell, like the gene gun method, electroporation method, micropipette method, and you must know about each of these methods at least in brief. Okay, then comes one more final slide of the genetic engineering steps. As we discussed, isolation of desired DNA, and here restriction endonuclease plays a role. Cutting of DNA at specific sites, cut source DNA to get gene of interest, cut vector DNA, formation of recombinant DNA. This portion is again very important. Amplification of gene of interest using PCR, that is polymerase chain reaction. Insertion of recombinant DNA into host cell, for which the host must be a competent one. Obtaining the foreign gene product, downstream processing. So following all these steps, genetic engineering is done. So here we have quickly discussed about the polymerase chain reaction. What are the components needed for the polymerase chain reaction? Basically DNA template, DNA polymerase enzyme, and 2 sets of primers. So I think you can go through this in more detail. The steps as well. So mostly these are the basic steps: initialization, denaturation, annealing, elongation, and final hold. So I think this again is important, so please go through all the steps in more detail. So this is just a quick recap video, so I'm not, because if I start going through the detail of all the topics, then I think we'll not be able to cover it ever. So just go through these topics. If you know about this, just revise it quickly. If you do not know, please go to the detailed video and check it out. Okay, let's move on to the next lesson, that is biotechnology applications. So here we will quickly touch upon transgenic animals, cryogenes, genetically engineered insulin, which is very important, biopiracy, ADA deficiency. So you also get questions from this deficiency. Okay. Now, the moment we talk about genetically modified organisms, so these are organisms whose genes have been altered by manipulation. So that's what we have been discussing in biotechnology as well. There are certain advantages of genetic modification. For example, it increases the nutritional value of food because we have the desired nutrients present in the food. Increased tolerance to abiotic stress, increased resistance to pests, because of which the plants might survive for a longer period of time. The productivity is also more. Increased efficiency of mineral usage by plants, so that in turn will help in the growth of plants. Decreased maturation time and increased yield. So these are the advantages of genetic modification. What about these advantages? Insertion of a gene can affect functioning of other genes in an organism. Absolutely. They can cause allergies. Food chain can be adversely affected. Disturbed ecology, because the moment food chain is disturbed, what is food chain? One organism depending upon other organism for food. Now, because of these genetic modification, it might happen that a gap gets created somewhere in the food chain, and if that happens, then the entire ecological balance is disturbed, because organisms which were dependent on that particular organisms will be adversely affected, which in turn will impact organisms which were dependent on these organisms now. So in that fashion, genetic modification can actually have can create disturbance in the ecology. So we often talk about the genetically engineered BT cotton. We say that it is more, resistant towards against pests. So what is BT? BT stands for Bacillus thuringiensis. So it is a bacteria that produces BT toxin. And what does this BT toxin do? It provides resistance against insects. So as a result, if you have BT cotton, because of the presence of this particular BT toxin, that entire plant gets resistant against the insects. So therefore, the plants will not get spoiled by the insects and it will survive for a longer period. So in BT cotton, how does the toxin work? The BT toxin is toxic insecticidal protein, and this protein exists as inactive protoxins in bacteria. Now, once these protoxins are ingested by the insects, they turn into active toxins. So otherwise, until and unless they are taken by the insects, they exist as inactive protoxins. But once they are ingested by the insects, they become active toxins. They bind to specific receptors in the gut of the insects. Gut wall breaks down and the insect dies. So basically, this toxin actually kills insects. So that ways it protects all the plants from the insects, and the plants lead a healthy life. Then comes genetically engineered insulin. Now, this is a very important application of biotechnology. So, if you look at the structure of insulin, it has a peptide chain A, peptide chain B, and disulfide bridges. So, if you see this blue line which you see, that is chain A, the green line is chain B, and these are the disulfide bridges which provide linkage between chain A and chain B. Now, insulin is synthesized as proinsulin, which gets processed to form fully mature insulin. So here this structure which you see is proinsulin. So you can see this blue portion is A, and then the green portion is B. And this gray portion that you see in between is C. Now, what happens when it is changed? When proinsulin is processed, it forms insulin, which is this, plus the C chain. So basically the C chain is removed from these two chains, and that's how we get insulin from proinsulin. So genetic engineering process to produce insulin. Bacteria cell is taken with plasmid DNA. Plasmid DNA is cut with restriction enzymes. Human gene responsible for insulin production is inserted into the plasmid DNA. Now, before we discuss about this, what is plasmid DNA? As I mentioned before also, small DNA molecule which is physically separated from the chromosomal DNA, and they can replicate independently. right? So here, this gene that you see, it is basically the human gene responsible for insulin production. So we take a piece of this gene and then we insert it into this plasmid DNA to form a recombinant DNA, and then we put this recombinant DNA into the body of the organisms to produce insulin. So as a result, recombinant DNA is formed, and this recombinant DNA is introduced into the new bacterium. So the bacterium multiplies, producing human insulin, as you can see here. Okay. Now, ADA deficiency, what is it? It is adenosine deaminase deficiency. Why does this deficiency occur? This occurs because the gene responsible for adenosine diaminase is deleted. Now, what is adenosine diaminase? This is an enzyme which is involved in purine metabolism. Now, the primary function of this enzyme is that it helps in the functioning of the immune system. And immune system is very important because immune system of our body protects our body against infection and other foreign substances, right? Now, this in adenosine deaminase, this particular thing, this enzyme helps in the proper functioning of immune system. Now, when there is a deficiency of ADA, then what happens? Then gene therapy is given to the patient. So, the first clinical gene therapy was given for ADA deficiency. So what was done in this gene therapy? A functional gene was inserted into an individual's cells to compensate for the non-functional gene, because the gene which was responsible for producing ADA was deleted. So that gene was non-functional. So instead of that gene, it was replaced with a functional gene so that ADA can be produced, and that's how this ADA deficiency was handled. So how can gene therapy cure ADA deficiency? Lymphocytes from patients' blood are extracted. What are lymphocytes? These are white blood cells with single round nucleus. They are part of the lymphatic system. And these lymphocytes are grown in culture outside the body. Functional ADA are introduced into the lymphocytes. The lymphocytes are transferred into the patient's blood. So that once we transfer functional ADA, what happens? Now, once the lymphocytes get inside, again they multiply and the ADA deficiency is removed. ELISA test, it is a diagnostic test for early detection of HIV. So, HIV AIDS is acquired immune deficiency syndrome. The pathogen which causes this is human immunodeficiency virus, which is abbreviated as HIV. This test is based on the principle of antigen-antibody interaction. So if you are already aware of antigen and antibodies, good enough. If not, please refer the detailed video to understand what are antigens, what are antibodies, and how do they interact with each other. Transgenic animals, these are animals with manipulated DNA. Now, why do they have manipulated DNA? Why do we even have the concept of transgenic animals? To study role of genes in diseases so that new treatments can evolve. Useful products are obtained from transgenic animals. For example, a transgenic cow gives more milk than that of a normal cow, because transgenic means we are trying to put more desired traits, more desired characteristics into that organism. So as a result, we get useful products out of transgenic animals. Can be used to test the safety of vaccines before being applied to humans, can be used to test toxicity of drugs. So basically, these transgenic animals, one advantage is that they give us useful products. The other advantage is that they can be used for experimentation, whether to experiment with drugs or to experiment with vaccines, but they can be used for experimentation. Biopiracy, what is it? Situation where bioresources are used by others for profit without proper permission from the people or countries. Now that's important because it is legally not allowed that without permission you use any of the living resources from a particular place or from a particular country for your own benefit, that too for your own monetary benefit. So biopiracy is that. So let's move ahead with the next lesson, that is organisms and population. The topics that we will quickly discuss under this lesson are short day, long day plants, regulators and conformers. This is very important from exam perspective. Homeotherms, poikilotherms, ectotherms, endotherms, age pyramids, Phases in logistic growth, this again is quite important. Mutualism, predation, parasitism, competition, competitive exclusion principle, and commensalism. Now, all of these relationships between organisms again is very, very important. So, let's get started. Short day plants. The name itself says short day. So they need shorter day and longer nights to flower. So they flower when exposed to long nights. They need minimum number of hours of darkness. So that means a minimum number of hours of darkness must be satisfied. It can be greater than that, but at least a specific number of hours of darkness must be there. Examples: coffee, chrysanthemum, strawberry. These are examples of short day plants. Just the opposite is long day plants. So they flower when they are exposed to short nights or long days. Examples are lettuce, spinach, potatoes. Day neutral plants. So their flowering does not depend upon exposure to light duration. So such examples are roses, cucumber, tomatoes. Okay, now let's talk about regulators, organisms which regulate. So mammals and birds have the ability to regulate, to adapt to their environment. Now what do we mean by regulate? That means as the environment outside changes, they also regulate the system inside their body to adapt with the environment. That is called regulate. And these types of organisms are called regulators. Now, these organisms can regulate in two ways. One is osmoregulation, the other is thermoregulation. Osmoregulation, they maintain homeostasis of organisms' water content. Now, what is homeostasis? Homeostasis is a state of stable equilibrium. So here when we are talking about water content, that means it is talking about a stable equilibrium of organisms' water content. What is thermoregulation? Maintenance of homeostasis of organisms' internal temperature. Thermo means heat. So the temperature has to be maintained. For example, during summers and winters, the external environment is very different. During summer, the external temperature is very high. During winters, the external temperature is very low. But organisms which undergo thermoregulation, they try to maintain the internal temperature of their body at a constant level, irrespective of changes in the external environment. That is called thermoregulation. So, in order to perform these regulations, there are, for example, for osmoregulation, we have gills present in fishes, or the kidneys in human beings, or the contractile vacuole in amoeba. So these are the organs in different organisms which help in osmoregulation. On the other hand, there are a certain group of organisms which conform. That means they do not have a mechanism to regulate their internal temperature and osmotic concentration. So basically, they are also called cold-blooded animals, or they are also called conformers. That means they do not change anything inside their body based on the environment. So examples are the reptiles, as you see, frog, lizards, crocodiles, they all come under cold-blooded animals. So if you do a comparison between regulators and conformers, you see that in regulators there is constant homeostasis, conformers little homeostasis. In regulators, internal temperature is independent of external temperature because internal temperature is always constant. Whereas in conformers, the internal temperature changes with external temperatures. Regulators consumes more energy because they are doing so much of regulation inside their body. The conformers consume less energy. Regulators are more active, conformers are less active. So this is how you can compare the two. Then let's talk about the types of animals based on how they control their body temperature. Homeotherms, they have a constant body temperature. Homo means same. So they basically fall under regulators category because they are regulating their body temperature. Poikilotherms, their body temperature adjusts depending on the environment. So body temperature changes with environment. So they fall under conformers category. Then we have endotherms. Endo means inside. So animals that obtain heat primarily from metabolic reactions that is taking place inside their body. So they are endotherms. They also fall under regulators category because they are obtaining the heat from within their body. So it is like their internal system. The examples of endotherms are birds and mammals. Ectotherms, animals that obtain heat primarily from the environment. So from outside, they are taking heat. So they are ectotherms. Ecto means outside, and they fall under conformers category. Examples are fishes, amphibians, and reptiles. Age pyramids. This is very important. Now, in every population, if we draw a structure based on the age distribution in that population, because every population will have young people, old people, very old people, infants. So, you will have people of various age groups. So, the structure that is formed when age distribution is plotted for a population, that is called age pyramid. So, here you see three types of pyramids. So, a pyramid of this type shows rapid growth, where you have a very, vast pre-reproductive phase. The black colored bar shows the pre-reproductive phase. This shows the reproductive phase, and this is the post-reproductive phase. Right? So what do we see? That as the age increases, the width or the length of each of the bar decreases. Correct? Now that is this type of graph shows rapid growth, whereas a graph like this shows slow growth, where you see the pre-reproductive phase and the reproductive phase, they do not have much difference. So it shows a slow growth, whereas a graph like this shows a declining population. So in case you are interested to learn in detail about each of these categories, you can refer the detailed video. But again, this is important. Now then you have the exponential growth model. So what is exponential growth? That means the population increases exponentially with time. Now this type of growth takes place only when the resources are adequate, and then only the population grows exponentially. Now in logistic growth, there are different phases. Now this one is again very important. So sometimes you get theoretical questions, sometimes you also get questions related to the graph from logistic growth. So there are three phases: lag phase, log phase, and stationary phase. Lag phase, that is the initial phase when there is a slow growth in the population. Then is the log phase, when there is a rapid growth in the population. And finally stationary phase, where the population growth rate decreases. That means the population almost becomes constant. So here the carrying capacity is reached. That means that is the maximum population that can be handled in that particular time. So these are the three phases in any logistic growth model. Now let's talk about relationships between various organisms. First we'll talk about mutualism. Mutualism is derived from the word mutual. So here both the species mutually benefit from each other. Let us take this example of buffalo and the birds. Now these birds help the buffalo by eating the insects which sit on the skin of the buffalo. So that's how the buffalo is benefited. And how are the birds benefited? They get their food, that is the insects, from the buffalo. So the bird is also getting benefited. Right? So both of them are getting benefited. So that is an example of mutualism. Similarly, if you look at honeybee and flower. So honeybee is getting the nectar from the flower. So the honeybee is getting benefited. How is the flower getting benefited? The honeybee is helping in pollination. So the flowers can reproduce. So the flower is also getting benefited. So these are examples of mutualism. Now one of the most famous examples of mutualism is lichen. So it is a symbiotic relationship between fungi and blue-green algae. Symbiosis again is the name given to a specific type of mutual relationship where two organisms are benefited from each other. So in this case, you see, this is the picture shows the lichen. So here algae, how is the algae benefited? So the algae gets protection from the fungi, whereas how does the fungi gets benefited? Algae is photosynthetic. It can prepare food by photosynthesis, and therefore the fungi receives food from the algae. So both are getting benefited from each other. Next is predation. Now species feeding on another species for their survival, that is called predation. For example, a lion feeding on a deer, so that's predation. Now series of organisms feeding on one another is called food chain. For example, plants eaten by the deer eaten by a lion, a tiger. So that's an example of a food chain. Now there are various defense mechanisms by prey species, because for example this tiger is the predator because it feeds on the deer. So in this case the tiger is the predator, whereas the deer is the prey. So there are various mechanisms which are followed by the deer so that it can protect itself from the predator. So animals try to protect themselves from predator. So one is like camouflage. For example, you would have seen that organisms which are, let's say, green in color, they try to hide behind the bushes so that they are not visible. Or sometimes even like here, you can see that the deer is trying to hide behind the trees so that the tiger is not able to see them. So there are various mechanisms which are used by the prey so that they can survive or they can protect themselves from predators. Next is parasitism, where organisms live on or within the body of other organisms, which are called hosts, and derive food from them. Now, most parasites harm their hosts, however, not all. For example, let's talk about the lice, which are seen in the hair of human beings. So, the lice gets its food from the human head, right, or from the human body, you can say. So the lice gets benefited. The lice also live on the body of the human being. So the human being is a host for the lice, and the lice is a parasite. Similarly, there are many tiny microorganisms which live inside the intestine. Some of them harm our body, some of them do not harm our body, but both of them are examples of parasites because they live inside the body of other organisms, that is human beings, which are hosts. Now, there are two types of parasites, ectoparasites and endoparasites. Ectoparasites, they feed on the external surface of the host. That means they do not live inside the body of the host. They live on the body of the host. For example, if you would have seen the weeds. So, weeds are present over other plants. However, they derive their nutrition from the other plant, but they are not they do not live inside another plants. They live on the surface of the other plants. Similarly, lice, they live on the surface of the human hair, not inside. Whereas endoparasites are those which live inside the body of the host. For example, the malarial parasite, they live inside our body. Or there are many other parasites which might live inside our intestines. So they are all examples of endoparasites. The word ecto always means outside and endo means inside. So that ways you can remember. Next is brood parasitism, organisms which depend on others to incubate their young ones. The best example is of crow and cuckoo. So the cuckoo, what they do, when they give birth to new ones, they come and keep them inside the nest of the crows. Now the crows incubate them for a considerable period of time. When they grow a little old, that time the cuckoo comes back and take their young ones. So they basically depend on other organisms to incubate their young ones. Next is competition, process in which fitness of one species is significantly lower in the presence of another species. So sometimes for the same resource, two organisms are like competing each other. So who will win? So obviously the more fitter species would win. So here comes the competitive exclusion principle, which states that two closely related species competing for same resources cannot coexist indefinitely. So over a period of time, the inferior one will be eliminated. So if both of them are, kind of fighting for the same resource. For example, if you remember the story of the Darwin's finches, you remember initially all those finches, they used to feed on seeds. But as their population started to increase, there was too much competition for the seeds. So what happened? A couple of them started eating insects. Right? So you either need to find out an alternative to survive. Otherwise, the competitively stronger one will survive and the inferior one will get eliminated. And then commensalism, where one organism is benefited while the other is neither harmed nor benefited. So the best example is that of the sea anemone and the clownfish. So you see the clownfish here, the sea anemone here. Now the sea anemone has tentacles, right? And those tentacles keep moving. So because of those moving tentacles, other organisms inside the sea do not come near the sea anemone. And the jellyfish, the clownfish takes advantage of that. So the clownfish always stay close to the anemone so that the tentacles of the anemone give protection to the clownfish as well and protect it against other predators. So that ways we can say that the clownfish gets benefited because of the sea anemone. However, the sea anemone is neither benefited by the clownfish nor is it harmed by the clownfish. So that means sea anemone is not impacted, but the clownfish is benefited. So this is an example of commensalism. So let's get started with the next chapter, that is ecosystem. So in ecosystem, we will quickly revise the important concepts from where you tend to get more questions. For example, if you actually analyze the more important topics or the more important parts of ecosystem, you would see that you do get around 2 to 3 questions from ecosystem. Sometimes you might even get 1 or 2 questions, but yeah, you will definitely not get more than 3 questions from ecosystem. So some of the important topics are net primary productivity, gross productivity, ecological succession, ecological pyramids, carbon cycles. So these are some of the important topics. So let's get started. What is ecosystem? Living organisms together with non-living organisms in an area form an ecosystem. Pond, forest, ocean, they are all ecosystem because they contain different types of living organisms plus the non-living components like air, water, soil, etc. Primary and secondary productivity, as I said, that this one is quite important, so please do understand it well. Primary productivity means the amount of biomass produced per unit area over a period of time by plants during photosynthesis. So it is the biomass produced by plants. So plants, which are the producers, they produce this much of biomass per unit area. That is called primary productivity, and it depends on the plant species of a particular area, nutrient availability, and environmental factors. So these are the factors on which it depends. Similarly, secondary productivity is the amount of biomass produced per unit area over a period of time by consumers. So primary productivity is produced by the producers, secondary productivity is produced by the consumers. So that's the difference between primary and secondary productivity. Next is gross and net primary productivity. Now in primary productivity also we have 2 categories, gross and net. Gross means the rate of biomass production during photosynthesis. Now a part of gross primary productivity, which is abbreviated as GPP, is utilized by the plants. Right? For what purpose? For respiration purpose. So some part of it is used by the plants and the remaining part is what we call as the net primary productivity. So it is the net rate of biomass production by plants excluding its utilization. So it is something like this, that let's say that I earn 100 rupees in a month, out of which I spend 10 rupees, or maybe let's say that out of 100 rupees I give 10 rupees as tax. Correct? So basically my net income, net take home, the net income that I am getting is 90 rupees. Correct? So 100 rupees is the gross and 90 rupees is the net. The same thing here. So the net primary productivity is equal to gross primary productivity minus R, where R is respiration losses. These are the utilization of the plants. Then you have the levels of consumers. So producers are the green plants which produce food by the process of photosynthesis. Then in consumers you have different levels like primary consumer which feed on plants, secondary consumer which feed on primary consumer, tertiary consumer feed on secondary consumer, then quaternary, and then you have decomposers, that is which feed on dead and decaying organisms. So basically you have 3 levels: producer, consumer, decomposer. In consumer you have various levels. Okay, now, and these different levels of producer, consumer and decomposers form a food chain. So you can have a food chain with 2 steps like this. You can have a food chain with 3 steps, 4 steps, 5 steps and so on. So here comes a very important law, that is Lindemann's law, which states that only 10% of the food energy is transferred from one trophic level to the next higher trophic level. Now each of these levels is called a trophic level in a food chain. Now as we go up the food chain, or as we go to the higher trophic level, at every level only 10% of the energy gets transferred. That means when the frog eats the grasshopper, only the 10% of the energy that was there in the grasshopper gets transferred to the frog. Similarly, when the snake eats the frog, only 10% of the energy that was there in the frog gets transferred to the snake, and so on. So only 10% of the energy gets transferred, and that is called Lindemann's 10% law. Then we have ecological pyramids, which represent food or energy relationships between organisms at different trophic levels of an ecosystem. So normally this kind of pyramid is said to be upright pyramid, where you have the producers, which is like a large base, and gradually as you go up to higher trophic levels, the number decreases. So you have huge number of producers, lesser number of primary consumers, even lesser number of secondary consumer, and then least number of tertiary consumers. Now, there are a few important points to be noted with regards to ecological pyramids, like the pyramids of number or biomass in most of the ecosystems are upright. However, it is not upright for all the cases. Sometimes it is inverted as well. And this is how an inverted ecological pyramid look like. Let us take this example. Let's say that you have this tree. There are insects which feed on these trees. Now, the number of insects are a lot more, and then there are birds which feed on these insects, and again the number of birds which feed on the insects are a lot more than that of the insects, right? So in this case it is an inverted pyramid. However, in most of the cases it is always an upright or erect pyramid. So pyramid of energy is always upright. So for energy, so we have learned about three types of ecological pyramids: pyramid of number, pyramid of biomass, pyramid of energy. So if you have forgotten them, please refer to the detailed video. Now, a given organism can occupy multiple trophic levels in a given ecosystem. Yes, that is very much possible, because if we are talking about a 5-step food chain, so there you might have a grasshopper at the second level. If we are talking about, let's say, a 4-step food chain, so the grasshopper might not be at the same level. It might be at a different level altogether. So a same organism can occupy multiple trophic levels in a given ecosystem. Then comes ecological succession, which tells that there is the gradual change in the composition or structure of species in a given area over time. And this gradual change over a very long period of time can bring about a huge change, a change such that a land which had huge amount of vegetation sometime, or which was a forest land, gradually got converted into desert, or something which was like, totally deserted, nothing was there. Over a long period of time, you see huge amount of vegetation in that area. So that is what is called ecological succession. So primary succession means process of succession starts in areas where no living organisms existed. So it is like the first step. Earlier than this, no living organisms existed, and now the living organisms started to exist. Secondary succession means process of succession starts in areas which lost all living organisms that existed there. That means before also living organisms existed there, but due to some natural calamity, maybe due to flood or any other natural calamity, all the living organisms ended up. And then a new succession started again. That is called secondary succession. Now, in plants, succession are of two types: xerarch and hydrarch. So xerarch succession starts in dry areas. So here it progresses from xeric to mesic conditions. Hydro means water, so it starts from wet areas and it progresses from hydric to mesic condition. So you see, this is hydric condition and this is mesic condition. So in case of xerarch, it progresses from this condition to this condition. In hydrarch, it progresses from this condition to this condition. So these are the two types of successions in plants. Now let's talk about carbon cycle. This is important. So please understand that you may be asked a question to describe the carbon cycle. So please go through it thoroughly. Similarly, for the phosphorus cycle, that how phosphorus or carbon gets circulated amongst the various components on the earth. Let's move on to the next lesson, that is environmental issues. So here some of the important topics that we are going to cover are air pollution and control, global warming, electrostatic precipitator, this is important, ozone layer, greenhouse effect, sewage treatment, biogas, and eutrophication. You must know about the measures taken to control air pollution. For example, anyways, you know what is air pollution, like presence of undesirable components in the air which make it harmful for us to even breathe. What causes air pollution, how can we control air pollution. So here we are discussing mostly about the control part, that is filtration of industrial pollutants before being released to the atmosphere, so that we have less pollutant thing coming out of the chimneys of industries and factories. Maintenance of automobiles to reduce pollutants emissions, so that again we do not have harmful gases coming out of automobiles. Noise reduction, maintaining cleanliness. So how do we filter the industrial pollutants? So that is where electrostatic precipitator or ESP helps. So it is a filtration device and it removes 99% of the particulate matter. It is very widely used in thermal power plants and industry. So you see, without ESP, it was like a black smoke coming out of the industries. With the help of ESP, the color itself is telling you that it has been filtered a lot. So how does it work? So it basically, what happens is, I mean, this is how the structure is more or less. So you have these collecting plates which collects all the particles which are like undesirable particles or smoke particles. So it acts like a high voltage electrodes which produce corona. So negatively charged electrons are released, smoke particles attached to negative charges. Net negatively charged smoke particles get attracted to positively charged collecting plate. So you see the collecting plate is positively charged. As a result, the clean smoke comes out. So this was dirty air and the dirty particles got collected here, so the clean air comes out from here. So anyways, you should understand this in more detail from the detailed video, that how exactly electrostatic precipitator work, because this is important. Okay, however, there are few limitations of electrostatic precipitator. For example, it cannot remove particulate matter of very, very small size, because it can only remove particles of considerable sizes. If you have very tiny particles, then it cannot remove. And however, these tiny particles can also cause different sort of respiratory disorders in human beings. Next, we'll talk about sewage treatment, because whenever we are talking about environmental issues, it becomes important to handle the sewage part smartly. So sewage needs to be treated in sewage treatment plant before disposal. Now, sewage treatment is carried out in two stages: primary treatment and secondary treatment. In primary treatment, the unwanted particles are physically removed from the sewage. There are two processes which help. One is filtration, the other is sedimentation. In filtration, what happens in filtration? Solid is separated from liquid by passing through a filter paper or by passing through anything which has pores. Sedimentation, what happens? The particles are removed from water under the influence of gravity. Like if you remember, if you put some sand in water and leave it for some time, what happens? You see that all the sand gets collected at the bottom and the clean water is on the top. So that's how you can separate the sand particles from water. So that is sedimentation. Now, in secondary treatment of sewage, the effluent from primary treatment is passed on for secondary treatment. Here, it is constant agitation of effluent happens, then air is pumped into the tank, vigorous growth of aerobic microbes. So, these aerobic microbes play a very important role in secondary treatment. These microbes consume the organic matter, and that's how it cleans up the sewage content, and that's how the sewage is treated. Secondary treatment is very important, so please go through it in more detail so that you understand the function and role of each and every part of secondary treatment of sewage. Now we'll talk about eutrophication. Eutrophication is aging of lakes, like how we human beings age. Right? Similarly, lakes also age. There are two types of aging. One is physical aging, the other is biological aging. Physical aging means the lake gets filled with more sediments. As a result, the lake becomes shallower. The depth decreases with time. That is physical aging. Biological aging means the old age lakes have higher primary productivity. Young lakes have lower primary productivity. So that's how we calculate the biological aging, that who has aged more. Now, when we talk about the physical aging, so eutrophication occurs what is the degree of eutrophication? Like how much the lake has aged? How do we know that? So that is known by the trophic states. There are three possible states: oligotrophic state, mesotrophic state, and eutrophic state. So oligotrophic state means little sediment. So this is your stage 1. When you have little sediment, therefore the depth is more. Mesotrophic stage, that is stage 2, where you have considerably more sediment and therefore the depth has decreased. And then you have eutrophic state, where you have maximum sediment and therefore minimum depth. And as you go down these states, you see that the primary productivity increases. That's because when you have more sediments, you will have more producers, more plants. Therefore, you will have more primary productivity. Now, accelerated eutrophication. Now, sometimes the lakes do not age naturally. It's like they suddenly undergo aging and suddenly the lake becomes old. So how does that accelerated eutrophication happen and what are its effects? So excess nutrient enrichment like nitrates and phosphates, because of which too much of plants grow, and as a result the sediment part increases. Growth of algae, lack of dissolved oxygen, and the harmful effect is that it is a threat to aquatic life, because the aquatic life will not be able to survive that well in an aged lake. right? So accelerated eutrophication normally happens because of human activities. So we should stop accelerated eutrophication and allow the water bodies to age naturally. So by now, all of us know what is greenhouse effect, right? So in greenhouse effect, what happens is the infrared heat which enters inside the Earth's surface, most of it is contained within the atmosphere. Only a small part it is able to escape. So as a result, what happens is the overall environment gets heated up, and this causes global warming, basically, right? So now in greenhouse effect, the most important greenhouse gases which causes this to happen are carbon dioxide and methane, right? So now this global warming, now what do we mean by global warming? The phenomenon that the average temperature of the earth is increasing day by day, that is called global warming. And what are the harmful effects? So if there is global warming, the glaciers would melt, mountains, ice sheets would melt, there would be a rise in sea level, increased precipitation, higher probability of flood, change in ecosystems. So everything is going to turn upside down. So therefore we always say that we need to put a check on global warming. Okay, and how can we put a check? By controlling the use of the greenhouse gases like carbon dioxide and methane. Okay, what are the causes of global warming? Increased use of fuels which release a lot of carbon dioxide, as I said. So carbon dioxide is a greenhouse gas. Second is deforestation. Third is overpopulation. Now they are all interlinked. When the population is more, their needs are more. They cut all the trees. Now when the trees are cut, what happens? The entire balance is disturbed, because when there are trees, they actually take up carbon dioxide, right? Which we release. But when there are no trees, there is nobody to take up carbon dioxide. So they are all interlinked. Ozone layer. Now, this ozone layer also play a very critical role. Now, ozone layer is an additional layer of ozone which doesn't allow the harmful ultraviolet radiation to enter inside the earth, because harmful ultraviolet radiation can cause diseases like skin cancer. However, these days it has been seen that there is decrease in amount of ozone in the atmosphere, which is causing depletion of the ozone layer. Now, the moment ozone layer gets depleted, ultraviolet radiation will be able to enter inside the earth's surface, and as a result, it will cause harm to all the human beings. What causes ozone layer? Use of chlorofluorocarbons, which are also known as CFCs. Now, these are released into the atmosphere in various applications like refrigeration, air conditioning, etc. So, we need to really control the production of chlorofluorocarbons so that the ozone layer doesn't get depleted further. What are the consequences of ozone layer depletion? Skin cancer, leukemia and breast cancer, damage immune system, snow blindness or cataract, damage plants. So basically there are a lot of health implications on human beings if the ozone layer gets depleted. That's because ultraviolet radiation will get exposed to us, or we will get exposed to ultraviolet radiation, which is very, very, very harmful for us. So what are the measures to control ozone layer depletion? Limited production of CFCs, minimize usage of pesticides, usage of eco-friendly household cleaning products, because all of these, they eventually release in the production of chlorofluorocarbons, and we have to stop that. Okay, now let's move ahead with the next lesson, that is enhancement in food production. So here we are going to talk about some important topics like desired characteristics of crops, dairy management practices, plant breeding techniques, moat, this is important, micropropagation and inbreeding depression. So when we talk about dairy farm management, what is it? So it deals with management of animals for milk and milk products production, which in turn are used for human consumption. Now, these dairy animals could be cows, buffaloes, camel, goat, sheep. Next is, how do we maintain quality of a dairy farm? It's important that we select good quality breeds with high milk yielding potential, because in order to meet the demand, we need huge amount of milk. So it is important that we breed good quality cows so that they are able to produce more milk. Proper feeding of the cattle so that they stay healthy. Cleanliness again so that they stay disease free. Disease resistant breeds. The breed should not have any diseases. In that case, the milk that they will produce might also have some diseases, and it can result in diseases in human beings. Regular visits by veterinary doctors so that they can do a regular checkup that the animal is healthy or not. There are two types of breeding: inbreeding and outbreeding. Inbreeding means mating between animals of the same breed. Outbreeding, mating between animals of different breeds. Now, why does this concept of breeding exist? Now, breeding obviously is needed for reproduction. Now, inbreeding and outbreeding concept came up because we wanted to produce desired traits in the new organism. So, for that, it is important that we mate two animals with desired traits, so that we are able to get a healthier, a superior animal. What is the concept of inbreeding? So closely related superior males and superior females are mated. Now, when we are mating superior males and superior females, it is quite obvious that the superior progeny will be obtained. The superior females produce more milk and superior males help to produce superior progeny. So we take the superior version of both male and female and mate them. That's the concept of inbreeding. Now, why inbreeding? That's because less desirable genes are eliminated, because we are actually selecting more desirable genes. Evolution of pure lines of an animal. Disadvantages: continued inbreeding reduces fertility. Correct. And productivity reduces. Now, when we actually inbreed, like when we actually mate similar organisms over and again, what happens is the fertility reduces over a period of time, and the overall productivity also decreases. Outbreeding. So in outbreeding, we breed a superior male and a superior female which are unrelated. Now, there are three types of outbreeding, that is outcrossing, crossbreeding, and interspecific hybridization. So outcrossing means animals from the same breed with no common ancestors. That means both of them belong to the same breed, but then they are not closely related. Crossbreeding means they are from different breeds. As you can see here, two different breeds of dogs are mated. And interspecific means you have two different species. So here you have a dog and the other one is a fox. So two different species, they are being mated so that you get a new organism with desired traits from both of these organisms. Another great example of interspecific hybridization is between the horse and the donkey. So when a superior male horse is mated with a superior female donkey, then what we get is a mule, which is a different organism. So this mule has certain characteristics of a horse, it has certain characteristics of a donkey. So it is neither a donkey nor a horse. So when we undergo interspecific hybridization, we get a new organism altogether. Okay, now there are certain controlled breeding experiments. For example, there is something called artificial insemination. So this is a technique in which semen from the male is injected into the female reproductive tract. So from any superior male, the semen is taken and it is injected into the female's reproductive tract. So this technique helps where normal matings are not possible. So let's say that if it is not possible to mate the female with the female, what we do, we just take the semen from the male and inject it into the female. Okay, there is another technique which is called moit. So in this technique, what happens? Female is given hormones for super ovulation, so that let's say that normally if a female gives 1 or 2 eggs, now when it is given special hormones, it will give multiple number of eggs. So as a result, what will happen? The productivity will increase with quality in short time. So this is basically to increase the production of superior organisms. So earlier if there were 2 eggs, so you will get 2 organisms, now you might get 12. So that is what is done in moit. So now when we talk about plant breeding, so there are certain steps that need to be followed. Now this one is again important. Sometimes in your board exams, you are asked to write down the steps of plant breeding. First is collection of variability, evaluation and selection of parents. So first you have to select which parents do you want, then cross hybridization, then selection of superior recombinants. After cross hybridization also you will get a lot of options. Out of that you have to select the superior recombinants, and then testing and release of new cultivars. So that's how plant breeding takes place. Micropropagation, it is a technique of producing a large number of plants in very short duration. So here what happens is every plant is identical to the original plant. So if you have this plant, so from this plant you produce huge number of clones, like exactly similar plants. Moving on to the next lesson, that is microbes and human welfare. So here we are going to talk about microbes in production of biogas, microbes producing acids, microbes as biocontrol agents. Now, where do microbes help? In a lot of places like household products, you look at yogurt or curd, so microbes play a role. You talk about baking, any bakery item, microbes play a role. Industrial products like beverages, antibiotics, enzymes and acids, sewage treatment, biogas production, control of pests and diseases, biofertilizers. So let's look at some of the microbes which produce some acids. So lactobacillus produces lactic acid, lactobacillus produce lactic acid, acetobacter aceti, acetic acid, clostridium produces butyric acid, aspergillus niger produces citric acid. So you should know these names because you may get a short answer question in your exam. Now, there are certain microbes which produce enzymes. For example, streptokinase is an enzyme, lipase, protease, pectinase. These are some of the enzymes which are again produced by different microbes. Again, you should know the functions of these enzymes and also the microbes which produce these enzymes. Next is microbes which produce alcohol. Saccharomyces cerevisiae. It produces alcohol, that is ethanol. Biogas. So what is biogas? It is a fuel gas which is mixture of various gases like methane, carbon dioxide, etc. It is a renewable source of energy resulting from biomass, and that is why it is so important, because it is a renewable source. It can be used over and again. It can be recycled, unlike the non-renewable sources of energy, right? So, and how do microbes help in biogas production? So these microbes which help in biogas production are called methanogens. These are anaerobic bacteria. They grow on anaerobic sludge during the sewage treatment, and they are also found in the stomach of cattle. So what you can do is you can actually go through the detailed process of biogas production, like in the biogas production tank, how exactly these microbes act, and you should know the entire process in more detail. This is how the biogas plant looks like. So you have a dome-like structure, the gas tank, you have a mixing tank, you have a digester, and then you have an outlet. So you should actually know the detailed, you should have a detailed understanding of the biogas plant. So I have already explained that in detail in the detailed video. The link of the detailed video is given in the description section. Okay, moving on to biocontrol agents. Now, biocontrol agents are natural enemies of insects, pests, or weeds. So there are certain microbes which are naturally enemies of all of these unwanted organisms. So they could be predators, pathogens, parasitoids, or herbivores. For example, some of the examples of predators are ladybugs, dragonflies, centipedes, spiders, cats, frogs, lizards. Parasitoid like Aphidius solemani, Eretmoceras, Encartia formosa. So these are all examples of parasitoids. That is, these are organisms which act as parasites. Then you have pathogens like in bacteria, again under fungi, under viruses. So these are all those organisms. I mean, these are disease-causing microorganisms, right? So they all act as biocontrol agents because the microbes can actually kill disease causing organisms, or they can kill organisms which are harmful for us. So you should know the names of all the biocontrol agents and how do they act as biocontrol agents, that is, against which organism do they act. You have biofertilizers where living organisms can enrich the soil quality. So they are called biofertilizers. Bacteria, fungi, cyanobacteria, they all act as biofertilizers also. So in biofertilizer, one example could be the symbiotic association between bacteria, rhizobium, and root nodules of leguminous plants. So this association helps to fix atmospheric nitrogen. So now the moment you fix nitrogen into the soil, you are increasing the fertility of the soil. So it is a biofertilizer. Similarly, the symbiotic association between fungi and certain plants, which is called mycorrhiza, is also it improves the soil fertility, because this fungi absorb phosphorus from the soil and pass it to the plant. It is also resistant to pathogens, better plant growth and development. So they also act as biofertilizers. Cyanobacteria also fix atmospheric nitrogen and add organic matter to the soil like Anabina and Nostoc. So you should know examples of all these bacteria or microbes which act as biofertilizers. You should know their specific names. And now we go to a very low priority lesson, that is biodiversity and conservation. So here we talk about just one or two important things. For example, how species diversity is related to area. When we say species diversity, it is about species richness or species evenness in the entire population. So we say that the relationship between the species diversity and the area is directly proportional, and it is also exponential. So we say that S is directly proportional to A to the power Z. Now if you take log on both sides, this is what you get. So this is the relationship between the species diversity and a particular area. Now, there are various causes of species extinction, because these days we see that a lot of species are becoming extinct, and therefore it is high time that we take care of them so that we can protect them from being endangered. So one cause could be loss of habitat due to deforestation. The forests are being cut down, so the animals are losing their habitat. Due to pollution, they get diseased. Human needs. So for human needs, human kill animals, human destroy their habitats. So as a result, we lose the animals. Now, overexploitation by humans. Overpopulation causes increased need of food and shelter, because of which humans, feed on animals a lot. So we lose animals once again. Invasion by alien species. So some other species come in. Let's say that this was a very peaceful area, habitat of small fishes. Suddenly a big fish from somewhere came up, and that big fish started eating up all the small fish. So what happened? These small fishes were exploited by some alien species which suddenly entered, acted as a predator or acted as a competitor, that they're competing for the same resource, and the alien species, being more superior, survived. Again, co-extinctions, associated species become extinct together. For example, host and parasite. So let's say that if the host becomes extinct, it is quite obvious that the parasite will also become extinct, because the parasite is dependent on the host for its survival. So how do we conserve biodiversity? There are two approaches: in situ conservation and ex situ conservation. In situ means on-site conservation. For example, national parks, wildlife sanctuaries, sacred groves, these are all examples of in situ conservation. That means we do not disturb the natural life of, or the natural habitat of the animals, and we still try to conserve them in that area. Hunting is prohibited, poaching is prohibited, nobody is allowed to disturb the animals, so we keep them in their natural habitat. The other is ex situ conservation, that is off-site conservation. For example, zoo, botanical gardens, which are not the natural habitat, but we create a natural habitat-like environment where the organisms can survive, and that's how we conserve them. So sometimes you may get questions on one of these techniques of conservation. Maybe you get question on sacred groves or biosphere reserves, or you may be asked to differentiate between in situ and ex situ conservation. So with this, we have reached towards the end of this video on Class 12 Biology Crash Course Part 2, and I hope that this video, Crash Course Biology Part 1 and Part 2 taken together, would have helped you to revise the entire syllabus of biology in a short period of time. So wish you all the very best for